Good day, ladies and gentlemen, and welcome to a novel in vitro method to predict cholestatic hepatotoxicity. All lines have been placed on listen-only mode, and the floor will be open for questions and comments following the presentation. If you should require assistance throughout the conference, please press *0 on your telephone keypad to reach a live operator. And if you'd like to ask a question during the presentation, simply type your question into the Q&A box located on the lower top left-hand side of your screen and hit the send button. And at this time, it is my pleasure to turn the floor over to your host, Graham Dick. Sir, the floor is yours.
Thank you, Daniel. This is Graham Dick. I lead commercialization at Qualyst Transporter Solutions. We at Qualyst provide in vitro hepatic models to help our clients predict the effects of drugs and other compounds on the human liver. We're pleased that you have joined our webinar about the C-DILI assay, a novel in vitro method to predict cholestatic hepatotoxicity. You've seen my name and email address in the invitation to the webinar and on instructions on joining. If you have any difficulties during the webinar, please send me an email. We'll try to address those. At this time, it's my privilege to introduce our speakers, Ken Brower and Jim McKinn. Dr. Ken Brower is Chief Science Officer at Qualyst and a co-inventor of the C-DILI assay. He has over 25 years' experience in preclinical drug development, leading research in metabolism, pharmacokinetics, toxicology, and absorption technologies. His research focus includes investigation of hepatic transporters. Ken will discuss hepatic mechanisms that underlie the C-DILI assay. Dr. Jim McKinn is founder and CEO of IonTox, a company providing in vitro toxicity screening services, custom research and development, and consulting services. He has over 25 years leading, advising, and consulting on research efforts in toxicology. His key areas of research have focused on understanding how chemicals affect key pathways controlling tissue health. In today's talk, Jim will discuss applications of the C-DILI assay. Ken and Jim have prepared remarks that will take about 40 or 45 minutes. We want to leave time for questions, so please type your questions in the Q&A box. And we'll see the questions here, and I'll be asking either Jim or Ken to address them. So at this point, it's my pleasure to turn the podium over to Ken. Take it away, Ken.
Okay. Thank you, Graham. Welcome, everyone. As Graham said, I'm here to talk to you today about our C-DILI assay for the prediction of cholestatic liver injury. As you know, drug-induced liver injury is one of the leading causes of acute liver failure in the U.S. It's also the #1 cause of regulatory actions in clinical trials and also in drug withdrawal. In addition to drugs, herbals and various dietary supplements are also important mediators of drug-induced liver injury. And we've known for quite some time that drug-induced liver injury may involve cholestasis and the accumulation of bile acids within the hepatocyte. And the hepatocytes also have the potential to adapt to these changes. And we at Qualyst feel that this adaptive response by the liver is an important component to capture when you're evaluating a compound's potential for cholestatic hepatotoxicity. One of the things that we wanted to do in development of this assay is to integrate various processes, so transport, metabolism, and regulation. We're focused on transporter-certified human hepatocytes, recognizing the species differences, especially in rats, between metabolism, transport, and regulation of bile acids. We're using a sandwich cultured configuration. And we also wanted a relatively short time frame for rapid turnaround. And Jim will talk more on these points later. And we also wanted a sensitive readout for that. Transporter-certified hepatocytes, we use and evaluate these. We evaluate lots of platable hepatocytes from various vendors for both uptake and efflux transporter function. And we look at a number of different things on this. It's important to note that out of the various lots of hepatocytes out there, only about half of those have sufficient transporter function in order to be certified. And we compare these to a database that we've generated from fresh primary human hepatocytes. And so these transporter certified hepatocytes are very important, not only when you're looking at transporter interactions, but also in induction and hepatotoxicity. In addition to transporter certified hepatocytes, it's critical to put those in the right type of system. And so we've recognized that generally just plated hepatocytes are not a polarized system. So in plated hepatocytes, or hepatocytes that are plated on a layer of collagen, the system is not polarized, there's no bile pocket formation, and the cannulicular transporters are internalized and not functional. And that's why the sandwich cultured hepatocyte system was developed. It's been well documented that when you put an additional layer of matrigel on top, that you reestablish cell polarity. You have functioning uptake and efflux transporters, and also the regulatory pathways are intact and functioning. And it's the only system that has a demonstrated in vitro to in vivo correlation in biliary excretion. Transporter function is very important, and this is getting to the difference between plated and sandwich cultured hepatocytes, as I mentioned, just as an example. For example, if you look at bile acid disposition, bile acids are taken up into the hepatocyte by NTCP and effluxed back into the cannulicular space through BSEPT. If, as in plated hepatocytes, that efflux transporter, BSEPT, is not there and functioning, you can get very high intracellular concentrations of bile acids, and this will lead to hepatotoxicity. I'm showing that here with CDCA as we increase the exposure of the hepatocyte to various concentrations of the bile acid. We see a very rapid increase in toxicity in this system. However, if we do the exact same study using the exact same lots of hepatocytes in a sandwich cultured system where the cannulicular efflux transporters are functioning, we do not get the high intracellular concentrations of bile acids, and as you can see here, you also do not see the toxicity with this system. So the use of transporter certified hepatocytes and a polarized system gives you the potential to have more predictive data. So cholestasis, by definition, is an impairment of bile acid flow, usually a decrease in bile acid flow, and this can happen through various mechanisms. And it can result in toxicity both in the liver, in the bile ducts, and in the systemic circulation. So there's multiple pathways for this. However, it's important to know that the liver routinely handles very high concentrations of bile acid. So there are protective mechanisms that the liver has. If we look at various patterns of clinical cholestatic hepatotoxicity, we can differentiate these into bile duct injury and also hepatocellular injury for this. And the bile duct injury is typically associated with immune reactivity. You can also have a mixed type of pattern, so the mechanisms may not always be clear. Our focus, since our system uses hepatocytes only, we're looking at hepatocellular injury in this assay. We and others have recognized that bile acids, in having the right mix of bile acids is critical. Human models are critical since rodents, as I mentioned, differ in synthesis, metabolism, and regulation of bile acids. We've optimized our system to produce in vivo relevant ratios of the primary bile acids, cholic acid and chenodeoxycholic acid, and their glycine and taurine conjugates for this. And in vivo, in humans, approximately 75% of the primary bile acids are conjugated to glycine. The rest are conjugated to taurine. And just as an example, in rats, you see very little glycine conjugation. Most of it is to taurine. So it's very important to keep in mind this species difference. And also, we know that bile acids differ in their transport properties and their cytotoxicity. And so a pool of bile acids better reflects the in vivo situation. Bile acid disposition and function. Bile acids are synthesized in the liver, and as I mentioned, we've optimized our system so that we see in vivo relevant ratios of these primary bile acids. Again, there are species differences. They're all excreted into the bile and then reabsorbed in the intestine, all by transporters, and returned to the liver via the portal circulation. Bile acids, of course, function in the digestion and absorption of fats and fat-soluble vitamins. But also, over the past years, it's become increasingly recognized that these bile acids are signaling agents for regulations of various processes in the body, including lipids and glucose, inflammation, drug metabolism, and detoxification. So these bile acids play many roles in the body. Bile acids are also very tightly regulated through FXR. And FXR activation will lead to an increase in FGF19, which will then lead to suppression of CYP7A1, the primary enzyme responsible for bile acid synthesis, and also induction of various transporters, including BSEP, MDR3, which is a phospholipid flippase, and the basolateral efflux transporter, OST alpha and beta. So as you can see, there's a number of different points that the disposition of bile acids can be impacted. And we focused in this assay on those that are primarily controlled by FXR. So that's cannulicular efflux through BSEP, basolateral efflux through OST alpha and beta, and also synthesis. It's important to note that all these other processes are also functioning in this model. They're just not modulated or affected by FXR regulation. So predicting drug-induced liver injury or cholestasis, historically, the concept was BSEP inhibition results in a buildup of bile acids, which are detergents and can dissolve cell membranes at high intracellular concentrations, leading to hepatotoxicity. However, as I mentioned, there's multiple ways that these bile acids can cause toxicity through various signaling events. And also, you can get direct cholestatic hepatotoxicity if you inhibit transport. So if you inhibit cannulicular transport into the bile, you can see increased bile acid concentrations, but also basolateral efflux transporters if you inhibit those. And also, we now know we need to take into account FXR activation for this. And again, it's important to remember that if you inhibit BSEP, the liver has a high ability to compensate, and we call that the adaptive response. So we feel that BSEP inhibition alone is not necessarily enough by itself. And so what I'm going to show here is a series of slides, but in response to high intracellular concentrations of bile acids, we see decreased expression of CYP7A1, so downregulation of that, and I've showed you that pathway through FXR. We also can see an induction of BSEPT, and I'm showing you here the different colors are different donors or different lots of cryopreserved human hepatocytes. And the variability here is represented, these were all done in triplicate, so you can get an idea of that. We're looking at the fold change in messenger RNA. And you can see as you increase the exposure to a bile acid, you get an increase in the expression of BSEPT for this. And also, you get an increase in the expression of OST alpha and beta for this. OST alpha and beta, it's a dimer, so you need both parts to function. So we see induction of both OST alpha and beta. So we can see this change in message. We can also link this change in message to changes in function. And here I'm showing a graph from a poster that was presented at the ITC workshop back in March. And using our sandwich cultured system and applying some mechanistic pharmacokinetic modeling, we can show that the cannulicular clearance, or the clearance from the hepatocyte into the bile, increases by twofold. We can show the basolateral efflux clearance, the clearance from the hepatocyte back into the blood, increases by approximately sixfold. And if you think about normally, in the normal state, about 75% of the clearance of bile acids is into the bile. About 25% is back into the blood. If you apply these fold changes to that, you see in the induced state, or after exposure to CDCA, the clearance into the bile is approximately equal to the basolateral efflux clearance. So you've really changed the disposition potential for that. In addition, we can link this effect to what's going on inside the hepatocyte. So I'm showing here the intracellular concentration of the total bile acid pool, and the blue is in the DMSO treatment, and so you don't see any change in that, which is what we would expect. But following treatment with CDCA, we see a very rapid decrease in the intracellular concentration of these bile acids. And that's due to two things: the inhibition of CYP7A1 and the increase in clearance both into the bile and back into the blood. And so this adaptive response can be initiated by several different processes. So if we just inhibit BSEPT, we can see this adaptive response. And I'm showing the adaptive response here by the fold change in OST beta messenger RNA content. And you can see as we increase the concentration of cyclosporin A, a potent BSEPT inhibitor, we can see this adaptive response come on board. We can also see this same adaptive response by adding bile acids to the system. And I'm showing here, if we add CDCA at 30 micromolar, and this is a non-toxic concentration, we can also see the adaptive response taking place. However, it's very interesting when you put all of these together. So now we have a little bit higher bile acids, but now we've inhibited the excretion of these bile acids into the bile. And you can see a very strong synergistic response in the FXR activation for this. The adaptive response, we've done a lot of work looking at how fast this happens. So here what I'm showing is the biliary excretion index, or the fraction of the bile acid that's excreted into the bile. I'm looking at GCA, which is the primary, is the most predominant bile acid in human hepatocytes. And we can see that following exposure to cyclosporin A, we see a very rapid decrease in the biliary excretion of GCA, which is consistent with inhibition of BSEPT for that. We also have done studies where we look in the same set of cells at the intracellular concentration of GCA, and we can see as we inhibit the biliary excretion of GCA, we increase the intracellular concentration of GCDCA. So we're blocking efflux into the bile. We see an increase in the intracellular concentration, and at some point that intracellular concentration gets high enough to begin to activate FXR, and that's what I'm showing in this one. So we inhibit BSEPT, we increase the intracellular concentration. At some point, we begin to bring on board this adaptive response. So, as I mentioned, you can affect intracellular concentrations by many ways. By inhibition of transport, but also you can impact it through antagonism of FXR. So again, what I'm showing here is the same thing I showed before. We can see this adaptive response with cyclosporin. We can also see it with CDCA, and then there's a synergistic effect when we have both together. However, now if we look at what happens when we have other compounds on board. So we look at triglydzone, which is a weak FXR antagonist, or DY268, which is a stronger FXR antagonist. We can see that we can block this adaptive response. So having an FXR antagonist, you can prevent the hepatocyte from responding to these higher intracellular concentrations of bile acids, and this will increase the potential for cholestatic hepatotoxicity. And so to look at cholestatic drug-induced liver injury, we really need to integrate multiple mechanisms. So bile acids are taken up into the hepatocyte and excreted into the bile. If we inhibit BSEPT, we will increase the intracellular concentration of bile acids, and at some point this will activate FXR and will bring down the intracellular concentrations of bile acids, or they'll reach some new steady state. However, if now we have a secondary insult, such as FXR antagonism, and or inhibition of basolateral efflux transport, we'll see those intracellular concentrations go way up, because now we've blocked all the pathways that the hepatocyte has to respond to that. And so compounds can increase the intracellular concentrations of these bile acids through BSEPT inhibition, but you need something else, and that can be either basolateral efflux inhibition and or FXR antagonism. And so to summarize all that, under normal conditions, high concentrations of bile acids are effectively handled by the hepatocyte. BSEPT inhibition may be an initiation event, but you need something else to happen. And then the severe toxicity is likely when you have a combination of these processes. And so what we're looking for in our assay, and Jim is going to be talking more on this, we wanted a physiologically relevant culture system. We wanted to integrate effects of parents and metabolites, and we wanted to incorporate multiple regulation mechanisms. And Jim, I will turn it over to you.
Great. Well, thank you, Ken. Great presentation as always, and welcome everybody. It's my pleasure to be able to talk to you about taking the amazing mechanistic data that has been developed around the human sandwich culture hepatocytes that really demonstrates the presence and function of these major pathways necessary for controlling bile acid homeostasis. And so the next question then is, how do you take this great mechanistic data and turn it into an assay that's relatively simple to use and straightforward and highly predictive? And a key component, of course, for success is that it is, in fact, mechanism-based. And whenever you have any type of an in vitro assay for predicting an adverse outcome that's mechanism-based, you have a much higher probability of predicting that event accurately. And as you've heard from Ken, and you'll hear again from me because it's so important, it's about the integrated whole cell model. You can't just take components out and look at them individually. They have to be in synchrony with one another. Ken's told you about the transporter certified hepatocyte, which is, I also believe, is a very important component of this system because it reduces variability between studies and also demonstrates that you have what you need to evaluate this model for bile acid toxicity. But also we need a single assay, we need a simple readout that is specific for bile acid-induced toxicity. And that readout should be relevant to what people are used to seeing in the clinic. So in the clinic, looking at liver enzyme leakage markers is a classical approach. And so in the in vitro model, we'll also be looking at a leakage marker. And of course, in order to be highly predictive, when you put all this together, the assay is very predictive for drug-induced liver injury through cholestasis, or the disruption of bile acid homeostasis. And one of the key things for developing any in vitro screen or testing model is to develop a good test set of molecules to demonstrate proof of concept. And so I'll be talking about that with about 45 different drugs. And when you put all that together, you have information that is very useful in terms of safety evaluation of not only drugs, but also chemicals in the environment. Now, a key component, as you've heard, is the fact that you have to have this a human model that's integrated. And integration here means that we have the major pathways present, both for the uptake of bile acids, but also the efflux pathways. And Ken has told you about BSEP in terms of efflux into the bile, as well as the MRP34 and OST for basolateral efflux back into systemic circulation, and the relationship between these key pathways and the FXR trigger, or the sensor for the need for this adaptive response. And cholestasis, I hope you can appreciate by now, is pretty complex in terms of its mechanisms of control. And when we talk about cholestasis, I mean, really I'm talking about bile acid homeostasis and regulation. And so this can be described in an adverse outcome pathway model, which is, these models are coming into vogue and are very nice because they express adverse outcomes as a mechanism of pathway-driven mechanism. So there's an initiating event, in this case, BSEP inhibition, and then there's a multitude of cellular events, which we've already shown you really represent the adaptive response of the hepatocyte that can lead to an adverse outcome, which is, in this case, cholestatic toxicity. Now, you can imagine from all that complexity, from all the things that Ken has told you, and from this adverse outcome pathway that I've just shown you, that if you try to predict cholestatic toxicity by only looking at BSEP inhibition, you could be missing some really important biology that would reduce the real predictive power of that assay. And this is kind of an example of that point. So this is work that Dawson did or published in 2012. It was extensive work with about 85 drugs where they used BSEP inhibition in inverted membrane vesicles from insect cells and developed IC50 values. And here you can see the different classes of toxicity in the liver along the X scale. And you can see here that there are many compounds where there's a weak association with BSEP. So as the IC50 value goes down, or we have a more potent BSEP inhibitor, there's toxicity that is observable. But there's also a fairly large number of drugs where the BSEP inhibition is really very high, so not a very potent BSEP inhibitor, and yet the toxicity is still present. So these would be examples of false negatives, because if you were using just BSEP to make this prediction, you'd say, well, these drugs have a very high BSEP IC50, so no toxicity. And of course, they do produce toxicity. And likewise, we can have drugs that are false positives. So here I mean that in the clinic, they are not associated with liver toxicity, but based on the BSEP assay, one might classify them as positive for cholestatic toxicity. So the idea is that there's a lot of biology that needs to be incorporated into the in vitro screen in order for it to be accurate. And here's another look at a subset of those same drugs from the Dawson work. Here's nine different drugs, and you'll see in the column here the IC50 for BSEP inhibition. And you can see the very low BSEP values, so very potent for several of these drugs. And yet when you look at the actual data, you can see that they really would have not predicted very well what's actually happening in the clinic. So these are, if you look at the clinical reports on these drugs in the liver tox databases, you'll see that these, in fact, would have predicted incorrectly. In fact, of the 9 drugs here in this small subset, only 2 predicted correctly. And if you do just some type of contingency analysis of this or Cooper's statistics, you can see it really is about a 50-50 proposition in terms of accurately predicting cholestatic liver injury based on BSEP inhibition alone. So if we go back to this adverse outcome pathway and its complexities, we can see now that we're incorporating BSEP inhibition. The model is monitoring or is capable of regulating bile acid accumulation. The model has taken into account FXR activation, which Ken has already shown you is very important in regulating and protecting the cells from bile acid accumulation. They can do that by inhibiting the synthesis, but also by inducing or increasing basolateral efflux, or increasing the safety valve, if you will, of the hepatocyte. And all of that leads to, gives you a much better model for predicting the toxicity that we're trying to predict. Now, if you look at that, and you might say to yourself, or be tempted to say, well, let's just take the components out and run in vitro assays on separate components. And really, that doesn't work very well either, because they're not integrated, they're not communicating with one another. And this is just a drug, indomethacin, where if you were sitting on a development project team in a pharmaceutical company or evaluating a tox profile for a chemical, and you had this data in front of you, it might not be too clear to understand. So here you've got a moderate BSEP inhibitor, not very potent, but you have also some effects on the basolateral efflux transporters, MRP and MR2 and 3, as well as a really potent inhibition of MRP4. And in addition to that, you also have an FXR antagonism, so a very potent antagonist or inhibitor of FXR. And most drug discovery teams also have some rat data, and in this case, you'd see that in the rat, you have hepatocellular necrosis. So in this situation, what call do you make? And of course, indomethacin is not related to that. So it's a conundrum. And so when we look at the in vitro models, then we just know that looking at individual components is not accurate because you need to have the communication between the components because of both positive and negative feedback that those components provide to the system. And you've got to have those compensatory mechanisms. So without that interaction, there's no adaptive response, so not very predictive. And the other reason is that in the animal models are often just not predictive of what's happening in humans. And I think Ken also alluded to this several times in his presentation, where he mentioned that in rats, bile acid constituents are different, transporter regulation is different, and bile acid metabolism is very different. So sometimes the rats just get it wrong. So the assay, as it stands today, features a 96-well format. It uses transporter certified hepatocytes, and it uses some very optimized culture conditions, so that we know that it takes 4 to 5 days to get those bile pockets formed up correctly and to get localization function of transporters. But in addition to that, it's in order to sensitize the cells, because remember, the functioning cell system has an adaptive capability. It's necessary to stress the cells enough so that they can respond to the toxicity of the drug. And to do that, we have to use a sensitization media called Qualyst, which sensitizes the cells by incorporating a mixture of lipids and bile acids that's has in vivo relevance. And when you use the optimized media and compare it to the standard media, now you can determine, you can differentiate between compounds that are producing a cholestatic type toxicity and those compounds that might be producing a direct toxicity or even a mixed type of toxicity. It's a 24-hour assay. It looks at both metabolites as well as the parent compound, and of course we're also looking at FXR expression changes, which are important. And it's a single assay readout. In this case, it's using a membrane leakage marker, LDH, and I'll talk more about that in just a moment. So we want to optimize the system, as I've explained. We've talked about the major pathways that are present, both the basolateral efflux pathways as well as the canalicular or bile efflux pathways, the uptake pathways through NTCP, and all of the pathways, many of these pathways, not all, but many are regulated by FXR. So that sensitization media is very important to allow the hepatocytes to respond to the potential cholestatic hepatotoxic effects of the compounds being tested. Now, this is an example of the assay and the different media and how they work. And on the y-axis, we have LDH decrease as the marker for toxicity. And we're looking at cyclosporin A, imatinib, and troglitazone in both media, the blue being the standard, and the red being the Qualyst sensitization media. And for cyclosporin A, you can see here that there really is no difference between the medias. This compound is not associated in clinical reports to cause cholestatic liver toxicity, even though it's a potent BSEP inhibitor. Over here on the far right, we see troglitazone, which Ken has also talked about a little bit. Here we see that in the standard media, there's no toxicity, but in the sensitization media, we have significant toxicity. And in the middle, we have imatinib, where we have an increase in toxicity in both media, but a much greater increase in the standard media, the blue, indicating that this is primarily a direct type of toxicity or possibly a mixed type of toxicity. So again, demonstrating the importance of using the different types of media. And again, looking at troglitazone and cyclosporin, I just wanted to point out the idea that this is really a threshold type of screen or model. And you can see here for troglitazone, as the concentrations increase, it goes really from a not much of an effect to a full effect pretty rapidly. Again, this is with the sensitization media, and with cyclosporin, we don't see any increase in the toxicity. The other thing you might be questioning or asking yourself about is why the differences in exposure concentrations. If you're comparing two drugs, wouldn't you want to use the same exposure concentration? And the reason for the differences is that when you have unknown drugs, you have to make an educated decision on what concentrations to test. And so to develop this assay, we based it on what we know about Cmax in systemic circulation relative to the Cmax that is observed in portal circulation for orally administered drugs. And we know that drugs that are administered orally have much higher concentrations in the portal system than in systemic. In fact, 10 to 20 times higher than the systemic Cmax. And so you can see for troglitazone, the Cmax, systemic Cmax is 6 to 8 micromolar. And so again, that 10 to 20-fold increase falls in the concentrations that were selected. And for cyclosporin, 0.5 to 0.8, again, justifying or helping to explain why we selected those concentrations for testing. And if it's very early or Cmax values are not known, then testing at a limit of solubility and downward works well also. So again, to remind you that troglitazone and its metabolites are BSEP inhibitors. They also can antagonize FXR. And as a result, when you have compounds that affect multiple pathways, you increase the risk of cholestatic toxicity. And remember that these alternate pathways include not only FXR antagonism, but also molecules that would inhibit basolateral efflux as well. So what was a rationale for choosing a test set of drugs to do a proof of concept evaluation or a test set? And initially we started with 38 drugs, and these were selected from the work done by Morgan and Dawson, where there is good BSEP inhibition data in the vesicle model. And then we selected exposure concentrations that are based on the differences in portal versus systemic concentration, which I just explained to you. And here are the 38 drugs that were tested on the x-axis. Again, looking at LDH as our marker for cholestasis. And we can use LDH as a marker for cholestasis because we've characterized this system. We know that the mechanisms for cholestatic toxicity exist, and it's been optimized using the sensitization media to respond to drugs that cause disruption of bile acid homeostasis. And when we do that and look at these compounds in both the sensitization media, the red bar, as well as the standard media in the blue, we can start to bin or categorize the molecules. So low toxic potential have LDH leakage less than 150% of control. Moderate potential is 150 to 200. And compounds that have a high potential would be greater than 200% LDH leakage relative to the controls. And so here you can see a molecule in the moderate to high here. Here's fluvastatin, and also ketoconazole here is highly toxic, and troglitazone's out here on the right. And before I move on, I just want to point out, too, that here in the middle we have imatinib, and this is the one where I showed you earlier where there was an increase with both media, but the greater increase was in the standard media. So this would be a compound that has direct toxicity or possibly a mixed type of toxicity. Okay, now we know that when we develop in vitro models that reproducibility is very important. And so here are hepatocytes from 3 lots or 3 donors. Again, looking at LDH leakage in the standard versus sensitization media. And the first thing you can see is that for cyclosporin, which are the first 3 sets here, there's no toxicity in either media. When you test troglitazone, we see a significant increase in the sensitization media only. And the magnitude of response is quite similar across the donors. And in fact, if you look at interplate variation or reproducibility within a single treatment, 3 to 9% is the coefficient of variation, and across all treatments, it's less than 6.5%. If we look at interplate across all treatments, less than 6% coefficient of variation. So again, the assay is highly reproducible. Now, I'd like to take you back to those 9 compounds I showed you in the beginning, that small subset. Again, remembering that using the BSEP inhibition in vesicles alone, you would have predicted, you would have made the prediction shown in the column to the right. And as I showed you before, almost all of those were incorrect relative to the clinical description of toxicity in the liver. The exception was triglycerol and ketoconazole. The correct predictions are shown here. Again, this is based on the clinical reports, which are shown in the next column. And those reports were taken from the liver tox database, and the emblem for that is shown at the bottom of the table. But when you apply the C-DILI assay to the same set of compounds, you have the molecules were correctly predicted, which is, to me, I realize it's a small set, only nine drugs, but it's 100% in the prediction power. Now, if you extend the proof of concept set from 38 to 45 drugs, which we've done now, and again, look at the contingency table of analysis, you can start seeing that we really have a very strong assay. So here we have a predicted value, the negative predicted value, the sensitivity or ability to predict something toxic, 87%, 87.5%, and specificity, the ability to correctly predict no toxicity, more than 97%. So obviously the accuracy here is going to be quite high, 96%, and this is with a respectable number of drugs now, 45 drugs that were selected from the extensive work done by Dawson and Morgan, where there was good human BSEP inhibition data and verifiable clinical liver toxicity. That's a key to these assessments. So the outputs and applications, this model, the C-DILI assay, can be used to predict potential for cholestatic hepatotoxicity resulting from high intracellular concentrations of bile acids caused by drugs. Because it's an integrated system, we reduce false positives and false negatives, and we've already talked quite a bit about the adaptive or compensatory response mechanisms, which are so important to have represented. And this can fit in screening, lead optimization or lead selection profiles in the pharmaceutical industry. It can be used to resolve concerns over false positives and negatives, maybe from a BSEP assay, and for regulatory submission. And in the environmental world, understanding how a chemical affects the liver, especially in terms in relation to an adverse outcome pathway, which the OECD is publishing now on a regular basis, it becomes a very valuable tool for assessing human risk. This is just kind of an outline of the different levels of assessment that you can do with the C-DILI assay. The threshold level shown at the bottom here is for looking at the potential for cholestasis, and you can do that. Using just the single sensitization media. You can contrast the direct versus cholestatic toxicity by looking at both media, the standard as well as the sensitization media, and then Qualyst is more than capable of helping you understand specifically which target or how your drug or compound is affecting the various components of the system. So then, to summarize, C-DILI is an assay that predicts cholestatic liver toxicity at the hepatocellular level. It's due to disruption of bile acid synthesis. It's a 24-hour assay in a 96-well format. It assesses both parent metabolites, and it can differentiate cholestatic from direct toxicity. As with every assay, there are limitations. This assay would not pick up things related to immune activity, bile duct blockages, or cholangiocyte damage. And there's possibility for false positives with molecules that cause a very high lipid accumulation, for example, amiodarone. So with that, I'll just thank you for your attendance, and I guess pass it back off to Graham for any questions that you may have. Thank you.
Thank you, Jim, and thank you, Ken. For everyone on the line, you can type questions into the box at the top left of your screen, and we'll see them here. We do have a few questions in here. And Ken, I'm going to address this question to you, and this is from Jim, who joined us in the audience. Is FXR antagonism involved in adaptive response?
Thanks. Yes, this system, as we said, integrates all of these potential regulation pathways or ways that the liver can handle excess bile acids. So not only upregulation of BSEP, which is an FXR mediated response, in addition, upregulation of basolateral efflux transporters, especially OST alpha and beta, which is again regulation, FXR regulated response, and also downregulation of CYP7A1 for this. So yes, FXR is intimately involved in the hepatocytes response to higher bile acid concentrations.
Thank you, Ken. There's a question that just came in from Nathan. Are the slides available? We've taken a video of the presentation today, and we will be posting the video shortly, and we'll send you a link to that video. Ken, another question for you. Can the Qualyst system detect cholestasis that's resulting from bile duct injury origin?
No to that, because the system only has hepatocytes in there, so it does not have any of the cell types associated with bile ducts. So that would be an injury that it would not be able to detect.
Thank you. For everyone on the line, you'll see we just advanced the slide, so you see my contact information as well as that of Ken and Jim. So you're free to copy that down and reach out to us offline as well. Let's see, another question here from Yvonne. So it's been demonstrated that compounds with dual BSEP and mitochondrial function inhibitors have high correlation to clinical hepatotoxicity. Do you also measure mitochondrial functions? It would seem that could be included during the 24-hour treatment period and would provide additional mechanistic information to strengthen the predictive model.
Yes, that's something we have thought of. It's not expressly looked at in this version or iteration of this assay, but it may be available in future ones.
Okay. Another question here is, for the assay time, why did you select 24 hours? Is that long enough?
Yes, so we didn't talk extensively about it here, but we've done a lot of internal research looking at the time frame of this adaptive response for that. And I did show a couple slides, especially where we're looking at the intracellular concentration of these bile acids after we've activated FXR, and probably 95% of the response to that occurs within the first 12 hours, and so that's a functional measurement for the adaptive response. If we look at things like messenger RNA changes in some of the transporters, notably BSEP or OST alpha and beta, or some of the regulatory factors like FGF19, we can see changes in that as early as 3 hours. So we based the 24-hour time frame on a functional change, and it would appear that most of that functional change occurs well before 24 hours. In fact, at 12 hours, it appears maximal.
Other questions from anyone? I know this brings us right to the end of the hour. Once again, I want to thank everyone for joining. And if you do have additional questions, please reach out to Ken or Jim or me. And we'll also send a note out to each of you who have joined us today and send you additional information. And we look forward to being in touch. So with that, any closing remarks, Ken or Jim, that you have?
No, not for me. Just thank everybody, and that's it.
All right. Thank you. Wishing everyone a great day, a great rest of your day. Thank you for joining. This concludes our webinar.